guys, I'm going to explain a question related to enzyme function in today's video. So let's begin. The question says the diagram shows the amino acids in a polypeptide. An enzyme catalyzes the hydrolysis of any peptide bond between a nonpolar amino acid and any polar amino acid. So the enzyme will catalyze the hydrolysis of any bond between this circle and any of these three shapes on the diagram, because this circle represents the nonpolar amino acid and these three shapes represents three types of polar amino acids. So how many small peptides and single amino acids will be formed by the action of this enzyme? So if we apply this information on the given diagram, we can see that this is a nonpolar amino acid and this is a polar amino acid. So the enzyme will catalyze the hydrolysis of the bond between them. So a small peptide will be formed on this side and another small peptide will be formed on the other side. And the same thing here. This is nonpolar and this is polar amino acid. So the enzyme will catalyze the hydrolysis of the bond between these two amino acids. And this will continue to happen till the end of the polypeptide. This will form small peptides, and also at some points in the polypeptide there will be single amino acids formed. And this is because if we can see this one, for example, there is a nonpolar amino acid here, and on this side there is a polar amino acid, and on the other side there is also a polar amino acid. So the enzyme will catalyze the hydrolysis of this bond and this bond. So here we will have a small peptide, and this one will be also another small peptide, and this will be a single amino acid formed. So if we count how many small peptides are formed and how many single amino acids are formed after we do this process, we will find that we have 13 small peptides and 4 single amino acids. So the correct answer will be D. This is it for today's video. See you in the next video.